Thank you very much, Ryan. Hopefully you can all hear me. My name is Steve Thomas from GSK. I'm going to take the audience today through my department's journey to find a suitable platform to store, search and share our metabolic knowledge. We want to be storing our data, searching information and leveraging knowledge about our molecules to make safer medicines. You've seen throughout Ryan's presentation the terms data, information and knowledge. And as I talk to people around my organization, I realize that some people use those terms indiscriminately and interchangeably. The analogy I use differentiates between them. Imagine you're walking down the street and across the road you spot somebody you went to school with. That's data, facial recognition. You recall his name. Information is the metadata you associate around that to make a bigger picture. You remember he's got 2 daughters and how well they're doing in school, and you remember the name of his dog. But you also recall that he's deadly dull, and so you escape into a shop to avoid him, and that's knowledge, something that you can leverage to make you choose a better course of direction. We want to leverage as much knowledge from our molecules so that we make ever safer ones for patients. Right. I'm from Ware's R&D site, about 20 miles north of London. Trading down here from R&D through platform technology and science. My department within drug metabolism and pharmacokinetics is biotransformation and drug disposition, what the body does to our drugs and where that happens. The body is completely ungrateful. It doesn't understand that the drug we're putting into it is trying to do it some good. It understands that it's not on its list of 3,500 to 5,000 chemicals that are supposed to be going around us every day, and so it gets hold of our nice drug and does some chemistry to it in order to excrete it through any route that involves a bathroom. That's where my department, structural ID, comes in so that we can work out the structures and find out whether that presents a potential safety threat. Coming on to structures and data, I'm going to show some structures and data now, but bear in mind that they've had to be changed and modified both to keep the lawyers happy and in some cases to make the points more clearly. So this is what we get up to. This is a trace of what became of our drug by LCMS in humans, and we've got a lot of peaks there for our human trace. What we want to find out is, did human beings produce anything that was particularly toxic or particularly different to any of our safety species? As you trade down there and look down to rats, you can see that there's a number of large differences between the human trace and the rat. Follow your eyes further down still, and you find that dog in this case is man's best friend, and all of the peaks that we see there in human are again found in the dog. So hopefully our dogs are then happy with taking our chemical, and we can then proceed carefully in human. But we have regulatory authorities, chiefly the American Food and Drug Administration, that will tell us quite how deep to dive into this data. One of the guidances that came out of the Food and Drug Administration was we must drop down to at least 10% of circulating parent. So looking across that trace at the top, if you ignore the broad humps at the end, which are likely to be lipophilic endogenous molecules, we've probably got about 12 compounds to work on there. But that works out if our main peak at 6.5 is our parent molecule. If the little peak at 9.3 is parent, we've got a much tougher job to do. So we've got a lot of chemicals there to work out and work out structures of. And we ought to be getting them right, because the implications of getting things wrong are severe. We're going back probably 8 years to the Par Excel disaster, where a company ran a clinical trial that went wrong, and 5 gentlemen had their heads swell up with a massive immune response. They lost digits, I believe they were all saved in the end, but the company that ran that clinical trial, or the company that made the drug, filed for insolvency 3 months later. So we don't want to be anywhere near that kind of problem. So there's some things on our side. The revenue from a successful drug gets piled back in to finance the best equipment, so we can have the safest drugs that we can possibly produce. Also, we tend to hang on to our staff. A number of my colleagues have got decades of experience in the field, and during that time they've generated a huge pile of historical data and gained a sixth sense to spot a structural alert, a chemical that might cause us some concern. But that data tends to go into a big dusty pile of paper, just like I've got on the left here. That's not just a graphic, that's probably what our archive looks like. So if our data in there is something that's going to help me with the analysis that I've got on my desk, I'm either going to need a very good memory or some luck or a colleague who happens to be looking over my shoulder who can help me. If the data is just in a dusty archive, I've probably not got access to it. So looking around the company, our US colleagues had historical use of ACDLabs software, where they would take the data for individual metabolites and store it away in a database. And the next compound they worked on, they would produce a database for that, and so on. Now that's useful as a storage department, but if I'm working on this structure here and I want to identify a metabolite, and it's the same metabolic change as was seen in this compound over here, I'm blind to it. There's no links between these databases. Can we find a way of linking these together?